Let's review topic 7, nucleic acids for high-level students. To start with, we need to review some of the experimental work that went into discovering the role of DNA and also its structure. So we'll start with Hershey Chase. And here's this concept of radioisotopes once again. So just like with Mendelssohn and Stahl and Kern's autoradiography, or even just diagnostic imaging, things like thyroid function, we see this idea of radioisotopes a lot in biology. So just making sure you're clear that they are versions of an element, an isotope, heavier atoms with unstable neutrons and protons in the nucleus. So that means they will decay or partially break down, giving off some of their excess energy. And there are actually several different ways they can do this. Maybe they emit positrons or gamma radiation, but for biology, for our purposes, as long as you know that the radioisotope is another version of an element that is going to give off some radiation that can be measured, that's why it's really useful in lots of biological applications. So physically, an isotope is different. It's going to be heavier, or radioisotopes specifically give off radiation. But chemically, in terms of the reactions they undergo, are the same as any other atom of that element. So that's why it's really useful for research. Here, we have in yellow, what they used in this experiment was a radioisotope of sulfur. So they used, what was that? It was 35, I think, 35 sulfur. So a slightly heavier isotope that breaks down a bit and gives off that radiation. And so they grew a bunch of these phages here, or you could call them a bacteriophage. And it's a type of virus. So remembering that viruses aren't living, it has no metabolism of its own. It has to use a host cell. It does have its own hereditary instructions. But before we understood if it was protein or DNA, this was a neat little experiment to determine that. So we can see here that this little phage is injecting something into the host cell, into the bacterium, to be able to make new viruses. So by putting sulfur in here, sulfur is a component of most proteins, so it's in some amino acids. So that made the protein component of the virus radioactive. So they grew them like this, so they would incorporate this radioactive sulfur, infected some bacteria, and then agitated that solution so that the little phage sort of coats, these shells, would come off. And would leave behind the hereditary information inside the bacteria. And then by centrifuging it, by whizzing it around, separating them based on density or mass here, they separated a pellet. This part here had the bacteria in it, and then any remaining bits of viral coat stayed here in the supernatant liquid on top. So they noticed then what they're showing by this bit that have been yellow is that when they measured for radioactivity, the radioactivity was all coming from the liquid. So that meant that the sulfur, the radioactive sulfur, which was in protein, stayed in the coats of the virus. Whereas when they used radioactive phosphorus, so the isotope 32P here, phosphorus, as we know, is part of the phosphate backbone of nucleic acids. So that's part of DNA. So they're able to choose radioisotopes that were exclusively found in protein and DNA to run this experiment. So this time, the little bacteria here are being infected by the phage. So it's injecting some information so that the bacteria will make new viruses. They give it a bit of time, again, agitate it all up or blend it up and put it into centrifuge. This time, they found that the radioactivity was in the pellet part where the bacteria are. So that meant that the thing that the phages were injecting into the bacteria must have been DNA and not protein. The DNA must have contained the instructions, must have been the unit of passing information on that genetic component. So that was some really strong evidence in the early days of trying to determine if protein sequences or DNA sequences were responsible for our genetics. And it might seem really obvious now, but if you back when you were looking at sort of possibilities, the genetic sequence having only 4 different nitrogenous bases as opposed to the 20-odd amino acids that we find in our cells in proteins, some people thought that proteins seemed more likely, but this experiment helped us determine that it was DNA. Other evidence to look at, Franklin and Wilkins. Now, this is the classic and very contentious photo, the X-ray crystallography or diffraction. Here we see that Rosalind Franklin took, and then was part of the research that helped Watson and Crick develop their model of DNA. So here, this is the actual photo. It's repeated a few times, and I must say I've taken this from BioNinja because I am not an expert in reading these diffraction patterns, and it takes a lot of training in mathematical analysis to really understand what's going on here. But what actually happens here is this sample, you fire some X-rays at it, and they will scatter off in different directions because of the interactions with the molecules. So diffraction just means like bending. So the way that they bend and scatter and form an image depends on sort of the placement of molecules, the charges on different groups and things like that. So here we get an image where there's a cross, which gives us some indication that there is a helix, and even how steep that is. So how many turns or how many bases we can see here, looking at, this regular, kind of getting some of that ladder shape, if you can kind of tell from here, and other things which I must say I'm not very good at interpreting, but looking at regions where perhaps there's water and also the nitrogenous elements and the phosphate components as well. So this helped give evidence for the double helix, because at one point they were thinking that might have been a triple helix or even more. Even the position of phosphate groups on the outside, which Franklin thought made sense anyway because of the hydrophobic nature of the nitrogenous bases and the more hydrophilic phosphates. So all of this helped build that greater picture. So it's a result of a lot of scientists' work that we have the current model we have today, not just the last people to finalize. All right. When we look at eukaryotic DNA, like in our own cells, we often think about genes and the versions of them, the alleles, but actually a good proportion of our DNA doesn't actually code for a polypeptide. There's some of the other things that your DNA might code for. It could be introns, which are parts of genes that end up getting spliced out in mature RNA, or they could be responsible for coding things, not RNA that would go on to make polypeptides, which messenger RNA is. It could be pRNA, the transfer RNA molecules, or ribosomal RNA, what ribosomes are made of. So that's an important part or function of your DNA. There's also heaps of regulatory regions that control gene expression. So you can have things like silencers and enhancers. These are parts usually upstream of a gene, and proteins that will they will bind to the DNA that will either encourage or stop the gene from being expressed. And there are other regions too that have structural functions. So that's centromere in the middle of a chromosome, or telomeres as well at the end. So here, for example, if you haven't, or to jog your memory, from the original gene here, when you first get RNA made, you make a copy in transcription, but before you get to translation, eukaryotic genes undergo this splicing to cut out the introns. So that's obviously a component that doesn't end up in the coding section. Here too, like our centromere is in the middle. This heterochromatin is this sort of these repeating sequences. And in fact, you might have seen something about the repeating sequences or heard it called satellite DNA before. So this sort of stuff in the middle here, we tend to think of as we call satellite DNA, or sometimes smaller repeating sequences might be micro or mini satellites. That's not really of any great concern to you, but what you do need to know is that there's something called tandem repeats. So that's sort of a more all-encompassing term for when we have base sequences that just go over and over again. Now, how that's useful, those tandem repeats are often actually where things like your ancestry, sort of genetic tests, what they might be looking at. Because some of those tandem repeats, some of those segments of DNA, like this picture here, you can see that GATA keeps getting repeated. There are a few different alleles that will control how many repeats a person will have in their non-coding DNA. So it's quite variable between different individuals. So that's a good thing to target if you want to do some genetic analysis, some DNA profiling, or maybe looking at family history, the kind of ancestry.com type things where they tell you are 20% from whatever country and so on. So if you think about what's happening too, like if you were to take, run some gel electrophoresis, and you were going to cut the DNA up with a restriction enzyme into fragments to run that electrophoresis, let's say you use a restriction enzyme that recognizes like this sequence here, this CTAGA or something, and maybe it makes a cut, I don't know, here, and there's one here, CTAGA. So you're going to have a cut all through these ones, but that one's going to be a bit further along, and that one. So these fragments then are actually different sizes. So the smaller fragments are going to travel further along your gel electrophoresis plate because they can get through that gel matrix, that kind of like a sieve effectively. That's how your gel is acting when the electric current is applied, and so we get an electric field and we have our DNA migrating along that gel, the small pieces go through more quickly. So that helps separate out and tell some genetic differences between people, and we can track some of those changes back through time in terms of human migration patterns. One of the other things we do with gene sequencing as well, so not necessarily tandem repeats here, but any component of looking at a genome and working out the base sequence, the Sanger method, which you don't have to call that, but that is the technique. And you would take from your sequence, remember that we have this polymerase chain reaction that we've looked at standard level, using the word amplify to say that we are replicating DNA to a high degree, and we're doing it in the laboratory using that TAC and polymerase operating at high temperature and not using helicase. Just remember that one. So you would provide in that mixture, you would have to provide lots of free deoxynucleotides in that mixture so that you can make those new strands of DNA. But what they do is they incorporate a couple of dideoxynucleotides. So they are different because there's a second oxygen that's removed from the sugar molecule, which means that when they join, they actually can't link to another nucleotide. So they stop the chain from growing any longer. And if each of these little dideoxynucleotides in the mixture, if each nitrogenous base, so A, T, C, G, if they are all tagged with a different fluorescent color, then you get something you can see in gel electrophoresis. And you can put them in order of size and work out the sequence of bases. So let's say here, for example, in this picture, the smallest fragment, let's say you started to copy this DNA sequence is in purple here, using the complementary bases. And then just by chance, you're going to get this whole range of fragment sizes. So let's say a little thymine here as a dideoxynucleotide with the red tag. That is going to only be one base long. So that would be the first one to make it to the end of your gel electrophoresis plate, or that would be the one that traveled the furthest. And then let's say you managed to copy a little bit of a longer sequence here and you got up to a C before you hit a dideoxynucleotide just by chance. And we've tagged all our C's green. We can see that will give us the next one. Or here we can tell that the next one closest to the end is a G. So that helps us tell us the sequence, then the next letter in the sequence must be G. The next letter in the sequence must be A, then another A, then a G, then a T, and so on. So we can read, or you can use a detector on a scanner sort of connected to a computer, because the amounts we're talking about are enormous. There, by the way, is that dideoxynucleotide. So we can see here there's an extra oxygen missing, which means that it can't continue to make a polynucleotide. It stops the growing chain. And then here's a picture of those like fluorescent tags and why you would want a computer to count those up and sequence that for you. If you haven't seen two, just because this here, you can see that nucleotides that are being added actually have 3 phosphates when they're added. So just to give you a little view of that, the extra 2 phosphates come off when it makes the new phosphodiester bond in the new polynucleotide chain. All right, let's look at now stages of replication in a little more detail for high level and some of the enzymes and proteins involved here. So here, this is DNA gyrase. Now that's focusing on this one here. And this one here is helicase. The helicase is the one that does the unwinding, separating the strands of your DNA so that you can make a new strand on either side that is complementary to the original. And it is that semi-conservative process. So this gyrase here, or in your textbook, they've just stuck with the name Proteome, which is accurate as well. But in your syllabus, they use gyrase. So I would do that out of preference for an exam answer. And that just helps stabilize it. If you imagine sort of if this section here has had to unwind, it kind of puts pressure on, you know, the remaining section to kind of wind up even more. So it's just a stabilizing molecule, really. And then if we focus, we've got this helicase. So we've got this DNA strand that's unwinding and we're moving along in that direction. And we would call this part here is fork or replication. We then need other little proteins that it just temporarily bind to help keep these strands apart. Otherwise, the strands would come together. They would reform those hydrogen bonds basically straight away. That's what the annealing is, be joining together. While they're being held apart then, we get a DNA primase. Now, this might seem a little counterintuitive. Is DNA primase because it's active on this DNA template. But what it puts there is the primer that it makes is actually made of RNA. So that is something that is a little bit confusing. The enzyme is DNA primase, and what it does, it puts an RNA primer here. It's a little starter sequence because polymerases cannot start a new strand from scratch. They have to add nucleotides to an existing polynucleotides. So we need these little primers. They're in green. So they're RNA, but they were made by the RNA primer made by DNA primase. Fusing. And then what we actually need to know too is it has a specific name, the polymerase. It is polymerase 3. Now, again, this is something your textbook actually doesn't address. Your textbook only talks about DNA polymerase in general terms, but your syllabus clearly states you need to know polymerases 1 and 3. Now, the numbers are not in order in which they're used. I think it's to do with the order of discovery, actually. And there are lots of different ones. There is a polymerase 2 as well, which we don't need to learn about. It seems to have kind of a proofreading role. And this is a model in E. coli, by the way. This is a prokaryotic model of DNA replication. It just starts to get too complicated in eukaryotes. In humans, for example, we have 14 known polymerases. So we'll just stick with the 2 that we need to learn for E. coli. Now, we've got these two strands here, and they're being replicated in different directions, at least to each other, because this DNA strand here is anti-parallel, and DNA polymerase 3 here has to work always in a 5' to 3' direction. Everything is 5 to 3. There's no trick question. It's always that way. 5' just means like the number 5 carbon on the deoxyribose molecule, and then 3', you know, that carbon, which we saw labeled on those little hexagon shapes. If you remember, like you've got oxygen at the top, and then you count your carbons, 1, 2, 3, 4, and then there's 1 up here. So when it makes that new phosphate group, makes that new backbone that it has to add to an existing 3 end, and then so we've got here like this 3 on this chain, and then that's going to be 5' anti-parallel on the new one. So that's the 5, and that's the 3. And the 3 that's sticking out, that's where the new nucleotide could be attached to. So this is also going 5' to 3' that way. But of course, our strand is opening up, this replication fork, sort of unzipping in that direction. So if we kind of try to see that moving, we can see this one here is continuous, and that one just has to do a little bit, a little bit, a little bit as it opens up. So this one is the leading strand, and that bottom one is the lagging. It's going to be a bit behind because it's always sort of on catch up. What we call these pieces here, these are our Okazaki fragments named after their discoverer, and they're just the segments of little bits of DNA. And we can see that you're going to need multiple RNA primers. So that DNA primase is going to be quite busy on the lagging strand. So there's not one shown here, but there would be one here. The next molecule, then we go to our other polymerase. Polymerase 1 gets rid of the primers and replaces them with DNA nucleotides, because remember our primers are made of RNA. So they have to be chopped out and replaced. And then our little fragments that are left have to be, these little nicks here have to be sealed with DNA ligase. So that makes a new phosphodiester bond here, a new covalent bond between our little nucleotides of our Okazaki fragments. So you might want to make for yourself a little sort of summary table, or just check that you're happy, you know, helicase unwinds, gyrase stabilizes, and so do proteins that keep the strands apart, so that primase can go in and make primers, and it's DNA primase for an RNA primer. DNA polymerase 3 lengthens the strand. DNA polymerase 1 gets rid of primers and replaces. And then DNA ligase is the one that connects fragments together on that lagging strand, which are our Okazaki fragments. Okay. Back to then looking at sort of larger structure in our chromosomes. So there's our DNA here. And we've seen before that it winds around histone proteins. So eukaryotic DNA does this. Prokaryotic DNA is considered naked. It isn't associated with proteins. But what we have here, we have these 8 proteins that are bundled together with 2 wraparounds of DNA. And there's this extra H1 here, this kind of handle on the outside that hold it together as well. So there are 9 proteins overall. And what that means is you have these kind of bundles that can then sort of coil up again on themselves. So we call this supercoiling. That we have this wrapping around, and then that bundles up, and then the whole fibre wraps up and condenses. So this actually, you know, it's quite a big structure then when we get to the level of a chromosome, which is why when they are about to undergo division cells, we can see chromosomes under a light microscope. So enormous, enormous mega molecules here. If we were to look at sort of some molecular modelling of what this is here, this part here would be like sort of an image of the histone proteins together, and this little H1, and then this part would be the ring of DNA around it, or trying to look at sort of 2 wraparounds of DNA double helix. Or we can see another kind of model here. This isn't showing the extra H1 histone, I'm sorry, from this view. But what we also need to appreciate is that these histone proteins can get modified, and that's going to affect gene expression. So if segments of that DNA can get copied into messenger RNA or not, and then converted into polypeptides and the like. Now we look particularly, it is possible also to have, by the way, but the ones we tend to focus on are methyl groups particularly. So normally when you have cell replication, you need to loosen these histones here and have some, well, this whole thing, sorry, by the way, that whole thing is a nucleosome. And so you need loosening between the histone proteins and the DNA to be able to copy the DNA. And you can have some chemical changes then. If you have little bits, so there's actually quite a few like little tails hanging out of amino acids from these proteins. You know, this is a very stylized little blob. And then depending if it's a methyl group, which is just CH3 here, and acetyl group's also got a double bonded oxygen in it. But depending on what's attached to those little amino acid tails, affects the interaction with DNA. Now, the methylation usually inhibits expression of the DNA. Not always. It will depend on what the amino acids are sticking out and which ones get methylated. But for the most part, the DNA, like the DNA here, is negative, and a lot of the residues of the amino acids are positive, like lysine, for example. Now, there's things like the acetyl groups, for example, just say they enhance transcription. Acetyl groups tend to kind of neutralize or cancel that positive charge, making the histones less attracted to the negative DNA. So that sort of loosens the coil, if you like, and allows those enzymes and transcription factors and things to get in and actually attach to the DNA and make little copies of genes, make messenger RNA to go out into the cytoplasm. Methylation, on the other hand, tends to tighten the coiling, tends to make those amino acids. But yeah, like I said, it does depend on which specific amino acid gets methylated. But at the end of the day, the key point here is that modifications to histone proteins affect how tightly it's bound to DNA and therefore how easily it can be copied. Now, that methylation process, or like changes that are happening there, the addition of small little chemical groups, it doesn't just happen to the proteins. We actually have DNA methylation as well. So it can happen at this level here on the DNA itself. So we have a whole variety of changes that can happen to the molecule at the sort of protein level or DNA level, or even entire chromosomes. Like in women, in females, you have 2 X chromosomes, but actually in early stages of embryonic development, one of those X chromosomes gets switched off. The entire chromosome is inactivated, which is quite incredible. And your bodies are a mosaic of like patches of cells where it's random which X chromosome is active, which is quite interesting, because when we looked at like Punnett squares and things and set-linked inheritance at standard level, we sort of treat it like sort of 2 copies of an allele, like an autosome, and we talk about things like colour blindness. Actually, realistically, women perhaps carrying like colour blindness, they actually have some of their cells in their retina that are colourblind and some that aren't. It's a patchwork of which X chromosome is active. So I think, but this whole study, this epigenetics, is of things that affect gene expression without changing the sequence of bases. So when I say DNA, but there are all these other sort of molecular changes that can happen to like chromosomes, to the DNA itself, and to the proteins that they're wound around, and that affects if those genes can actually be copied and expressed then in the cell. Now, when you have an epigenetic change, when you have some methylation of DNA or some histones, that's actually really important, because that's part of your signaling process for your cells, and it helps them differentiate. Start out as those totipotent stem cells and then become pluripotent and multipotent, and they change and they become more and more differentiated. We see that these epigenetic changes are kept. So when a cell divides and undergoes mitosis, we still have the same changes already made, so that they don't revert back to being those original stem cells. Now, usually when we get to the point of meiosis, so forming gametes, there's this reprogramming that occurs. All these epigenetic tags are removed so that they're not passed to the next generation. We actually find in about sort of 1% of cases they are maintained, or there are some epigenetic things, and we're talking about changes in the environment, even things like stress, or what you eat, or exposure to other chemicals perhaps that you're breathing in. They can be affecting you in utero, so before you're even born. And we're seeing now that actually we've underestimated in the past what things can actually get passed on. There are some sort of fascinating and very sad kind of examples of people who've been through traumatic events, and their grandchildren are still predisposed to sort of perhaps mental illness or certain responses to stress because of that. So it really is quite an interesting and expanding field of research, because you kind of always thought that in the past you wipe the slate clean and you make a new individual and you don't have any of these changes. And when we talk about evolution, we kind of down talk Lamarck, the guy who thought that the giraffe just kind of stretched its neck a little bit, then its baby had a longer neck, because we didn't think that, that nature versus nurture argument, we didn't think the environment or our experiences would affect our genetics, but we're seeing now actually perhaps it does, at least to a small degree. Other things that control our gene expression. So again, I keep saying gene expression, and by that I mean a gene gets copied and then translated into a polypeptide. So whether or not that happens also depends on things that are, those non-coding parts of our DNA that I mentioned before. So here's just a little picture, by the way. This part here is really the gene. Now there's open reading frame. That's not something that I've seen on your syllabus, but it is something I've seen on an exam question before. They've said something about, and they've used the term open reading frame on the phrase. So just so you're aware, they're talking about base sequencing. But you might have a gene here. So of course, this is going to be ATG, the first triplet, because the complementary, so this would be the sense strand here, and the template strand would have complementary bases. And then when that is transcribed to messenger RNA, that would be making AUG, of course, because we get uracil in RNA, which is our start codon for methionine. And then we get to a stop, which tells us to stop making our gene product as well. And then here upstream, we might have a region called a promoter. That's where RNA polymerase is originally going to attach. Now, this time we're talking about RNA polymerase, not DNA, because when we're expressing a gene, we make short segments of messenger RNA. We're not replicating the whole chromosome here. We're just going for a particular gene that we want expressed in the cell. There's a promoter region where this RNA polymerase can attach. There are also regions that we would call silencers and enhancers, and as the name suggests, silencers stop gene expression and enhancers promote it. So I just put that over the top. There's a lot going on in these pictures here, but we can see like polymerase here, these large molecules. You see this tartar box here, just this sort of start sequence that's common to eukaryotic genes. And there are other things, we say promoter proximal elements, like things that are just not far away from the start of that gene and the promoter that might help our polymerase attach. Things like here we've got an enhancer where a protein will come in and enhance that polymerase attaching and copying our gene that we want. Or we have a silencer that acts as an inhibitor when the protein is attached. And some of these proteins that could be coming in are things like our hormones. So if I just cross back to what we've looked at recently in option D, remembering like steroid hormones, for example, are those lipid-based ones that can get through the cell membrane and they get into the nucleus and actually act right on the DNA and affect our gene expression. So there'll be this little hormone receptor complex here, and that will be a protein that acts on a silencer or enhancer region of the DNA. So it'll make this little complex here. So that means the hormone, what it's acting as then is a transcription factor to affect the transcription of that gene. Or if you remember too that, these examples here, like our vitamin D or our cortisol, some of them act as repressors and some of them act as activators here. So here, this repressing or silencing kind of region is going to stop transcription. Or here, it's going to activate, it's going to promote it and help perhaps that RNA polymerase to assemble on our gene here to make our messenger RNA. And just as a refresher too, to contrast that, if you have protein hormones, they don't cross the membrane. They trigger secondary messenger, which acts at the protein level for cellular responses. So it's only the lipid hormones that are ones that get into, like steroid-based ones, that get into the nucleus and affect that gene expression. Now, stages of transcription. So we've got our DNA here. Remember, we're not talking about replication anymore, copying the whole lot. We're just talking about making a little copy out of RNA instead to go out into the cell and be active. So we have different enzymes. We have RNA polymerase, and actually, we don't need a whole bunch of enzymes for this one, but some of the other principles are the same. So we still have this 5 to 3 direction. That's true for everything. But our RNA polymerase is able to initiate transcription by unwinding a little segment of our DNA, separates it, and then it moves along in that 5 to 3 direction, adding new RNA nucleotides. And then it will reach a region known as the terminator, not Arnold Schwarzenegger, but a region that causes it to kind of buckle and detach, and it will come off. Now, in our DNA, because we are eukaryotic, we actually make a primary transcript, or the first piece of RNA that's made actually gets modified before it goes out and gets translated. So we have these things I mentioned before. We've got these exons and introns get cut out, and exons the express region, and they will get spliced together, and then they can go out and get translated. And there's this concept too of alternative splicing. That means that actually there's not always the same exons that get put together. We can have different combinations, which gives rise to a lot more variation from our genome. So our proteome is a collection of like all the polypeptides and proteins in our cells, is much larger than our genome, because we can actually chop and change the genes we've got to make more than one product. So that's as well, and something that you need to know. So here's another image, just if you need it. So we've got this initiation, we've got a site, a promoter region, our RNA polymerase over here. It moves along, it elongates and builds our new messenger RNA molecule, and then it comes to a region where it detaches, terminates. And that RNA processing, we can have that splicing of different exons, and then we also add a little cap onto our messenger RNA. Called a 5' cap because of where it is, the 5' end, which is just some phosphates really, and a poly A tail, a bunch of the nitrogenous base adenine. Now, not all of it gets translated. So again, that thing about coding and non-coding, we even have these regions here that end up, sort of getting lopped off later on, or that help regulate the process then of translation. So let's kind of build that into the picture. We've got our DNA, we've got some messenger RNA made, and it gets processed. So our introns are spliced out, and we get a little tail, cap and tail added, and then it goes out through a nuclear pore, and then we have to hit a ribosome and some transfer RNA molecules with our amino acids, and that's our process of translation. Now, of course, this is specific to eukaryotes. We have all our translation occurring in the cytoplasm, whereas in a prokaryote, it's still a cytoplasm, but it doesn't have to exit the nucleus first because there's no nucleus. So you can actually be having the process of transcription and translation simultaneously in a prokaryote, which you absolutely cannot in a eukaryote. It has to leave the nucleus first in between transcription and then translation. So our ribosomes, let's have a little look at those some more. So they come together, a little hamburger or sandwich on our messenger RNA. The bottom unit comes in first, and we have these sites as well. So if you remember this EPA, I do because I just think of the Environmental Protection Authority, but this is the one that comes in first. Well, actually, the very first one just comes straight into the middle, but then all your other tRNA molecules have an amino acid site, so where they bring the amino acid. Just remember A for amino acid, P for the polypeptide chain where it's growing, and then E for exit, which is pretty straightforward. And of course, small and large subunits are not very hard to tell from a picture. And we feed through like our little kind of reading a strip of information here, our messenger RNA. So translation then, we have the steps actually called the same thing, this initiation, elongation, and termination, which is nice. If you remember one, helps jog your memory, but it also means you can get them quite confused. So it might help you to make sort of some comparison tables of what's going on in each. But that initiation here for translation, for making a polypeptide now, our messenger RNA that's mature has to bind to the ribosome. So a small bit comes on first. There's our little small subunit that attaches to a messenger RNA. And it goes along that 5 to 3 direction until it hits a start codon. So it finds that AUG. And then it's going to the tRNA is going to come in, and the tRNA has to have a complementary anticodon. So try to use that kind of terminology. because they're all worth marks in an exam answer, or you might not get the point without it. And then our large subunit will come on in as well. So that's now initiated. By the way, this little GTP is a bit like ATP, but with guanosine instead of adenosine. So a different nitrogenous base, but it's got 3 phosphates still. So a triphosphate, and it's also energy rich, but it's specific just to these reactions. So we've got there our initiation, and then it's going to move along. If I can get the next image to come up. So our first one, well, actually this isn't the first now because our polypeptide tells us that we've had 4 come in. But our new amino acid here goes into that A site, tRNA, carrying our amino acid, brings it into place in that A site, and the P site is where that polypeptide chain is growing. And so we've got to have this catalyzed reaction here from using this GTP. And then it's transferred and we move, they all sort of shuffle along, go across one spot so that this tRNA exits, and then it can be reused in the cytoplasm. So there's some of our stages there, elongation. Let's get rid of those markings, or they'll stay there. And then we have termination. So a release factor is not a tRNA molecule, but it's still got an anticodon. It's got a match of codon here for the stop codon, but it's of course not carrying amino acid. So then our polypeptide is free, and our ribosome disassembles and can be reused again as well. Now, if we focus on these little tRNA molecules for a second, here they are here in this kind of clover shape, with those little hydrogen bonds here holding them together. Now, this part here has to have the anticodon, that has to have 3 bases that are complementary to the codon, a messenger RNA, and then we have a site here where the amino acid attaches. It's a little acceptor loop here. Now, if I just change that, then I'll pick that image and we talk about the amino acid attaching. Attaching here, that requires some ATP to join, so it is an energy demanding process, and it also requires an enzyme. Now, because we've got 20 amino acids, we actually need 20 enzymes. So there's a different enzyme for each amino acid because they're so specific. That enzyme substrate specificity is something that's kind of applied here, or a good example. So it requires energy, it requires a specific tRNA activating enzyme, and then the energy is actually then used because it's a high energy bond, that amino acid that's attached, and that will be transferred when it makes a peptide bond to the neighbouring amino acid when it's brought together in the ribosome and catalysed there to make our new polypeptide. Now, this process of translation, of making polypeptides from messenger RNA, can happen like a big factory line here with lots of ribosomes all working on one messenger RNA molecule. So we can have lots of polypeptides. It can be quite efficient. So here, this is an electron micrograph showing that. So these little guys are the ribosomes. That's a growing polypeptide chain here, all these bits, and that's our messenger RNA through the middle. Now, the ribosomes, they can be free in the cytoplasm, and for the most part in our cell, they are ATS ones, but don't forget in mitochondria or in chloroplasts and plant cells, they're 70S like prokaryotes, that endosymbiotic theory. And proteins or polypeptides that are made by free ribosomes are generally intended for use within that cell. They don't require any modification or packaging or anything to export them. Whereas the ones that are made in this rough endoplasmic reticulum do tend to be for export. So we can see here we've got some DNA in the middle. We'll have a segment that's unwound to make a little messenger RNA copy, and that's going to come out into our rough endoplasmic reticulum, where we can see we have our ribosomes hard at work, churning out our polypeptides into this flattened sort of bit of vesicle here. And that might bud off and go to Golgi where some other chemical reactions are occurring to perhaps add a carbohydrate group or something else. a prosthetic group, something inorganic maybe, and then these vesicles might then go for exocytosis here, all different things. And then what's controlling gene expression could be, say, a steroid hormone that's come in, it's passed through the membrane and in here, and that's triggering gene expression by perhaps coupling with another protein to make a transcription factor. So it all comes together. All right, finally, we'll look at our protein structure. So we've looked at then DNA replication using those enzymes to make whole new sets of DNA before cells divide, and then making a small copy of just a gene, making messenger RNA instead is transcription, and then translating it into our polypeptide. And then what happens from our amino acids sequence, so that's our primary structure, the order in which the amino acids are assembled, that is going to dictate everything else. That's our first level of structure, and what order they're in. And then some of these things here, like this is the acid part and the amine part. Now they're linked to their neighbours by peptide bond, but they can actually affect things that are opposite them. So you might have like a little interaction happening sort of between here, you know, holding together a little helix, or between going across perhaps and holding some pleats together. So you're going to have slightly different sort of mechanisms of folding, and it will depend what the sequence of amino acids is, so that there are these little groups sort of poking out near each other, and they're going to interact, the amine and carboxylic acid groups. And they're held together and stabilized then by hydrogen bonds. So that gives us our secondary structure here. And then our tertiary structure, and this is important, especially in our globular proteins. Sometimes fibrous proteins sort of only get to about here, because they're long, as the name suggests, fibrous. But to be globular, then you've got to have additional interactions that help it give it more three-dimensional shape. And that's usually the side chains and the amino acids. So there's R over here, what's hanging off the side of our backbone. And some of them, you know, like we mentioned right at the start with Hershey Chase, that a lot of amino acids, or some amino acids, have sulfur in them, that most proteins have sulfur. So disulfide bridges are quite a strong bond that can form. Or we can have things, other ionic bonds, or even just interactions like hydrophobic regions. So groups that are attached on the amino acids that don't mix well with water will all try to hide in the middle here of our globular protein. So they'll all try to face in, and that might help our protein fold up as well. And then not all proteins will have quaternary structure, but plenty of them do, where perhaps multiple polypeptides come together, or something else is added, like a carbohydrate or that inorganic prosthetic group. We call it a conjugated protein when you have that prosthetic group, like, for example, the iron heme group in hemoglobin. And just a quick look then, if you're interested, for that tertiary structure and those interactions, we can see of all these amino acids, they all have this amine and carboxylic acid. Actually, they're shown as zwitterions here, where the hydrogen has moved from here to here to have an extra one. It's fine. But it's these side chains that are shaded slightly darker that give us all those interactions. So some of them are polar, and they will interact with each other, or the non-polar tend to be those hydrophobic ones that don't want to interact with water, whereas these polar ones will move to the outside. And then if they've got charged regions on them, perhaps another carboxylic acid group, for example, and they will attract each other as well. And I think, where have we got here? Like there's a sulfur, for example, some sulfur on some of these. We might be getting like those disulfide bridges. All right, that's all for topic 7. That's a lot. I don't know if you might need to watch it again or pause or go back over something, but please do ask if you have questions, because it's a pretty full-on topic, and it does help, I think, to look at the processes side by side, just perhaps make some tables of the key steps or the key enzymes that are used in replication, transcription, and translation, so that you can clearly distinguish between them, because it is really common that students get confused in an exam and apply their knowledge from one of those processes to the other, and then they miss the point of the question and they don't get any marks. So it is really important to be distinct on those. Once you know them, they are good questions to answer, because they have sequential steps and quite a lot of technical information So the mark schemes usually have a lot more possible points you could get for the marks. There's quite a lot of options that the examiner can choose from when they're going through and giving your work. So they are good questions to attempt if you're on top of it.